so with this diagram what we have is our DNA molecule, our primary or pre-RNA transcript, and then we have our final mRNA transcripts. And you can see that we have alternatives. We have alternative mRNA transcripts. One is going to produce one gene, the other an entirely different gene. The gene is actually the same, sorry, entirely different protein, but the gene is the same. So something happens after transcription, our post-transcriptional modification, that produces this difference. So our primary RNA transcript has all of the exons, all three blues, the green and the purple. But then we have down here, after RNA splicing, that we only have the green or that we only have the purple. This is going to produce obviously a very different protein. So if we just use a really simple example, let's say that the green exon produces a functional protein in the eye that makes up the lens. And this is how we're supposed to have our eyes working, so that, that we're not short-sighted, that we don't have any vision problems. But on the right we have our purple protein, which creates a problem. It means that we are, let's just say that we are short-sighted. Now, for whatever reason, that splice occurs and we end up with the purple splice, or the purple transcript. And so we end up with the dodgy crystal, the dodgy protein. This is kind of how a lot of these things work. We can think of, particularly it's interesting when we have environmental factors that change things. And that's going to make, we're going to talk a little bit about environmental factors and how they manipulate things, particularly in Year 12. You'll have some opportunity to do some research or potentially do some research around that for your SHE task.